To go back, I guess it's the red button. Morning all, my name is David Hunt. I'm one of the genetics consultants from Southampton. I've been involved with PURA syndrome for over a decade now, and I've been asked to speak to you this morning about the patient registry. So I've deviated from the title in the programme to talk about past, present and future. There we go. So, what I will talk to you about is why natural history studies or patient registries are so important for rare disease and why we have to embrace these, because as a community they really are the way of driving research forward. I will spend a little bit of time talking about the existing patient registry, which is based at the University of Southampton, how the recruitment has panned out over the past few years since that's been running, what we've learned from this experience and why the decision was made to transition to a new platform. I'll talk a little bit about what we plan to do to repurpose the registry in Southampton, which will remain operational, and what we will do with that data. So why are natural history studies so important? Well, it comes down to the fact that rare diseases are, on the whole, poorly understood, especially very soon after they are first described. And this is because when clinicians report those first cases, there's often a bias towards a specific type of presentation. So the individuals who are involved in those discovery papers tend to be individuals who are more severely affected. So over time, what we tend to learn is that actually the spectrum of disease is far broader than was first described in the literature. It's also important to note that the vast majority of genetic testing takes place in children, and often very young children. So there is a bias towards descriptions of children and very little in the literature about how affected adults present. So, on this point, what I have got here, just as a demonstration, is all of those published papers to date about PURA syndrome are shown in orange, and you can see that of the 227 patients in the published literature whose ages are clearly stated, the vast majority are in childhood. And in blue, we have the individuals who are in the registry. So this is shown as a percentage by age up to the age of 40. So if we just focus on children, we can see that in the published literature, they make up 92% of the descriptions. In the patient registry in Southampton, they make up 78% of the enrolled participants. So this here really is the big unknown. We've got quite a nice picture, a nice understanding of what PURA syndrome typically looks like in childhood. We don't have a very good understanding of what it looks like in adulthood. But as you can see, the benefit of a registry is that over time you start to accrue really important data, because you're going back to the same patients year after year to get up-to-date information and to really build the phenotype, the natural history of a disease. Published literature is static. It's a description of a child or a young adult at one point in time, and it may not necessarily be updated. But the purpose of a registry is to continually update. So that's what helps us to build a much better understanding of the long-term prognosis, the life expectancy, what problems may be on the horizon. And I think the other thing that's really important to state is that although we very quickly get a good idea of the common clinical features in rare disease, and you can get a good sense of PURA syndrome just from the first 3 or 4 publications, what becomes much harder is working out the rare manifestations of a rare disease. And the analogy that I use when I meet with patients in outpatient clinic is that if you wanted a good representation of mankind, you wouldn't settle on a sample of 20 or 30 humans. That wouldn't be a good representation, especially of health problems. We would barely capture common human health conditions in such a small sample. The problem with rare disease is that we're dealing with small patient numbers. So when we have ultra-rare diseases and there are only a few dozen people who have been diagnosed in the whole world, sure, we have a sense of the core features, but we don't really have any idea about the less common or rare manifestations of disease. PURA syndrome is now sufficiently well diagnosed that with in excess of 800 known cases around the world, we ought to get a much better sense of the less common presentations of PURA syndrome. So what sort of things are enriched in this condition against the general population? And that is the sort of thing that we can only really identify through centralized studies and registries. And so this is all part of what we would call deep phenotyping. So why are registries so important? Well, they also give an opportunity to families to voice what their concerns are, what the day-to-day quality of life issues are, what the care needs are for them. And ultimately, when you meet with a clinician in an outpatient clinic, you're talking specifically about the medical problems, the management of a condition. You're not talking about the sorts of things that you need in terms of support on a day-to-day basis. So in terms of building a better understanding of what sort of things would make a tangible difference, actually having patient-reported sort of outcomes and feedback is really important, and these are collected through natural history studies. And of course, when we have really solid collections of data, they can help influence management guidelines. And these are ever so important for rare disease because we try to standardize recommendations, and that is part of what the Gene Reviews talk is about this afternoon. And one other thing which registries are particularly good at is helping to determine whether there are genotype-phenotype correlations. So what that means in plain English is, if you have a specific type of genetic change in the PURA gene, does it mean that you are more likely to have a particular clinical presentation? So do certain types of genetic changes have a greater risk of developing seizures or having issues with bone health? And at the moment, we don't know because we don't have a large enough sample of patients. But with technically access to hundreds of patients with really good quality data, it should be possible to determine whether there are any statistically significant differences and whether there are any clear associations between the exact genetic change and the clinical presentation. And of course, thinking about potential therapies, it's important to note that biotechnology and pharmaceutical companies obviously are looking to develop therapies to help in the management of rare diseases, and they have technically hundreds, if not thousands, of rare diseases that they could decide to target. Those which are more attractive tend to be those that have larger populations that could benefit from a therapy, and also those that have fairly solid natural history studies or patient registries. It's really helpful to those companies to know how many patients there are, what type of genetic changes they've got, what the geographic distribution is, what the demographics are in terms of ages of those patients. So I think that this is an important development in terms of making sure that the PURA syndrome community has the appeal for investment of biotech and pharmaceutical companies. And of course, if there is a good patient registry, then it can potentially help to access clinical trials, knowing where patients are, again, what their ages are, what type of problems they have, will be able to determine the centres that perhaps ought to be running clinical trials. So way back when we first started thinking about establishing a patient registry, which was in 2016 when we had the first annual conference, we were thinking about the size of the PURA syndrome community, which was about 100 patients around the world who had been diagnosed at that time. And of course, there wasn't much money. The PURA Syndrome Foundation was only just being set up at the time. And we had to think about how we could start a registry and do that on a shoestring. And actually, quite a lot of goodwill went into this. There were some private donations, there was some funding from the PURA Syndrome Foundation, and also some support from the Clinical Informatics Research Unit at the University of Southampton in effectively repurposing an existing clinical trials platform to become what is the Patient Registry at the University of Southampton. So that went through the standard ethical approval process and was officially launched in January of 2022. It's compliant with European data protection regulations. And as I say, it's based at the Clinical Informatics Research Unit at Southampton. So how does it work? So the registry works by patients being granted, or families being granted, access to the online platform. All of the study documents and consent documents are embedded within there. And ultimately, once somebody has been given access, they can start to create a profile and complete the consent process if they choose to join the patient registry. So in numbers, this has sort of plateaued over the past few years, because last year we had decided that actually it was time to look for an alternative to the registry in Southampton. I think it's fair to say that accrual has been slower than we had expected, and I talked about this at the 2024 conference. So as of June 2026, there are 121 users who were granted access who actually logged into the site, 112 enrolled participant profiles. Of those, 4 didn't complete any demographic information. And you can see there that over the past year it's completely plateaued, but it was actually a fairly steady rate of accrual over the years that it was running. So looking at the modules within, so apart from the demographics module, there are then 8 clinical modules. And you can see that we have an overall completion rate of 63% as of June 2026. And this is a global registry. There are people from all around the world. So 21 different countries are represented over 5 continents. By far the lion's share of participants come from the USA, as is represented in the heat map. But what are the challenges that we face? Why did we have problems with accrual? So the issue here, and this is a slide taken from 2024, is that we had a very low conversion rate. So at that time, in June of 2024, we'd had 192 requests to join, but of those, only 42 turned into complete data sets of surveys. So, that low rate of conversion was a big problem. So we had to work out why a third of patients never even logged in, or families, after being granted access to the registry, and why a third of patients who had logged in didn't actually generate a profile. And of those who did generate a profile, why they didn't necessarily go on to complete all of the questionnaires within. So the main barriers, I think it's fair to say, the biggest one is the user interface. It is a research platform that is typically used by research trials, clinical trials assistants and research nurses, and we had no way of changing the user interface. So when you were given access to the registry, it was accompanied by a 17-page step-by-step illustrated user guide, which is a little bit off-putting, let's be fair. So it's a little bit overwhelming to be given so much information just to get to some surveys to provide some data. The other thing is that you could only access it through desktop computing, so no slick mobile apps to do on the go. You'd have to sit down at a laptop or a desktop to actually be able to do this in a meaningful way. Another key point is that it was restricted to the English language, and we did try, with Dirk's help, to basically translate the user guides, but we couldn't actually translate the interface, so it was purely in English, which obviously is limiting the number of families who would be comfortable entering data. And then, of course, time constraints. We're asking people to sit down and fill out very long questionnaires. And I think another key issue is that there was very limited real-time support. You had to basically send an email to our research nurse who would only respond within limited UK working hours, and of course it may not be a response that comes back immediately, so from raising a question it could be a few days or a week later before you actually get a response. So moving forward, what have we decided to do? Well, as Liz and Eva have outlined, the plan is to migrate to a serviced off-the-shelf platform that supports many rare disease communities. They have a very slick user interface, and hopefully some of those problems that we encountered with our effort at the University of Southampton will be sort of bypassed through this new offering. Ultimately, the study in Southampton will continue to operate, but with a more restricted mandate. So we will be supporting the biobank, helping ensure that samples that are submitted here in Munich have accompanying phenotypic information. And the plan is also to use the platform to gather clinical data from enrolled patients locally, so that we can still use this as a natural history study, but not necessarily populated with data from parents, but with data from clinicians like myself who are seeing PURA patients every year or two and keep them under long-term follow-up. So if you have submitted data, it remains securely stored. Ultimately, what we can do with the existing data is somewhat limited because, unfortunately, as you saw, some of it is incomplete, it's not iterative, so we may only have a single snapshot in time for a lot of that data, and of course it is not clinically corroborated. We haven't gone back to families, clinicians to make sure that the data that we've been given is correct and to obtain supplementary data. But there is still some really valuable patient-reported data that can be used and should be mined. In terms of transferring data to the new platform, we need to explore how that might work in practice. It's unlikely that we're going to be able to just transfer all of the data in bulk, because we can only transfer data according to our research ethics approval in an anonymized format. So we'll need to explore how that could work in practice and whether we need to go back to each and every family individual to get consent to export data, and whether it could go directly to the new platform provider or whether it would have to go back to families first. I don't know. That is something that we have to work out. So I would like to acknowledge all of those who've been involved to date. So it's quite a big team at CIRU. The PURA Syndrome Foundation, of course, has supported this over the years. And I'd like to thank all of those who've made donations as well to help the registry along its way. And there are a few of the key people who were involved over the years in setting up the registry and supporting it day-to-day operationally. Thanks.
So while we are waiting for the presentation, I'd like to say, I should start it? Yes. Okay. No, this was the second one. I would like to go back. Yeah. I'd like to say good morning to everybody. And I really like to thank you for the opportunity to talk a little bit about biobanking. I'm Bettina Lorenz-Depireux, at the moment heading the Helmholtz-Munich Biobank, and I'm also head of a small research group focusing on biobanking data and ELSI, and ELSI is the abbreviation for ethical, legal, social issues or applications. On my slide, under the title, you can find 4 very important people. Most of them, hopefully, or pretty knows them. it's Dirk. He's really also you know that he's also working at the Helmholtz Center. And it's Simone. I'd like to thank the foundation because they also finance part of Simone's position. Simone is the biobanking organizer, and she's sitting right to this room here. And then there's also Katrin, and she's also with me. Katrin is the leading technician within my lab at the Helmholtz Center. What I'd like to talk about is what is biobanking, and at the end of the talk, hopefully everybody here is an expert in biobanking. This is how it looks like. This is just a view of how the Helmholtz Center Munich looks like. We are approximately here at the moment. And the biobank is here. And hopefully I will see many of you tomorrow at the tour for the biobank. And if you look more into detail, the campus of the Helmholtz Center Munich is approximately 10 to 15 kilometers in the northern part of the city. It looks like this. There's nothing very special. It looks like a black box. That's the biobank. And Melissa is laughing because she was already there. This is what we are doing. We are coming from, or let's say the biobank is located at the Institute of Epidemiology. So usually we are collecting cohorts, what means that we've got many of samples, and as you can see here are just the icons of German cohorts we are taking care of. At the end, at the moment, we've got over 1 million samples. For biobank, it's not a small biobank, but it's not a big biobank. The big biobank is really close to us. We are just the small sister. There is also an own building for the German national cohort, and they've got over 200,000 participants and over 25 million samples. But we are right in the middle, focusing and coming from the cohort part. What's interesting is that these cohort, this is the oldest cohort in Germany, because there we've got participants and they went to their study center for over 40 years. So this is just a collection, and then they will be examined again and again. This is how cohort looks like. There's one study center in Augsburg, and they are doing many different kinds of examinations, questionnaires, biosampling, and at the end they are all stored within the Helmholtz Munich Biobank. This is just an example. Concerning I don't know if there was one young guy and he's really a soccer fan, so I think that's pretty interesting. These are our global players within the biobank. And on the right-hand side, you can see the team Data and Elsie, and on the left-hand side, you can see the team Biobank. And we are working also with the German Centers of Health Research, different ones, the ones focusing on cardiovascular diseases and the one also focusing on infectious diseases. So we were also the first biobank associated with collecting the national pandemic cohort for COVID-19, started in 2020. But there's a soccer player missing here, and I hope that someone can help me. And this is the new child on the block since last year. You've got an old biobank. So the biobank of the PURA syndrome is also hosted here in the biobank of the Helmholtz Center. We are also member of European organizations like the BBMRA ERIC, where all the biobanks within the European Union are connected, who are interested to being part of this. I'd like that you know a little bit things about biobanking. So that's nothing new, it's pretty old. Even in the former days, they started collecting seeds, or you know that they are also starting collect animals, and unfortunately not every sample will reach the biobank, what means gets the ticket for example, the Archenoa. So probably this is why we missed the unicorns, and we don't have unicorns anymore. At the end, we try to organize everything in a very structured way, and I hope I can show you this tomorrow. You're collecting where your special kind of samples, and at the end you have to put in somewhere into, let's say, a big drawer, but you have to find it in this drawer. That's the most important thing. And so the first step, as we are doing on this other room here, is the collecting biometallurgical material, human material, non-human material. You can store what you want within a biobank. We store it under professional standards, what means we've got a certification, ISO 9001. This is a global certification. The researchers, they are usually using then the samples, and their samples are for studies for the whole population or for rare diseases, for example, the PURA syndrome. And at the end, they are using these for the different kinds of research projects, and I'm sure you will hear about these things in the afternoon then. This is how it looks like. This is exactly what they are doing on the, in the room here behind me. There you can find a form first, because everything is like a protocol. You have to write down everything, and maybe you can see it. The most important thing is, when you're registered in the registry, as David already showed, then you will get a number, and at the then what means is that you replace your name by a combination of numbers and letters. We call this pseudonymization. So the biobank won't get any data from you which are clinical data or identifiable data. The biobank will only get this code for the sample sample ID. And then we are collecting, we say it always, sample associated data. These are data like when was the blood drawn, when was the blood taken, when we are going into the processing of the blood samples, and at the end, when the samples will be freezed. So time to centrifugate and time to freeze. These are biobank associated data. And we start here on the left corner, it's the blood sample. And after you put it into a centrifuge, you can divide the different parts of the blood. What means you can see then there's this yellow liquid, and this is, for example, plasma. And then there's something in the middle, there are a few cells, and the red ones, these are the cells. The red cells, most of we are not interested in, because they don't have DNA, and nobody's collecting. We are not collecting the red thing. We are collecting the yellow thing here. And what is the biobank doing? It's processing, making aliquots from the plasma. You can do this by hand, you can do this by automation. So we've got a robotic, but we've also got a technician. It just depends on how much of the plasma you got, how standardized or harmonized this is. And at the end, it looks like in this box. I will show you or give you the vials and the box tomorrow, that you can see it and you can really take it. It looks a little bit different. Each of this aliquot has now a barcode, or it's a 2D barcode, and now this is the identifier. And you can put everything now into a computer system. And this is why we've got these big part data, and we call it Laboratory Information Management System. This yellow box, you can store it. Then it depends on which kind of biosamples you have. You can store it at -80. You can also store it in the gas phase of the liquid nitrogen. so with minus 140 degrees Celsius. And at the end, if you've got an application from one of the researchers, should go through a use and access process, or and then you should get such MTA, material transfer agreements, then we can put out the aliquot and send it to the user. So I don't want to go into detail, but most important is that there's many IT and data work behind, but there's also a quality control work behind. And this is how it works. On the upper side, the first thing what we have to check is that there is an informed consent, that there is all these ethical parts. There we have to be careful with the protocols, and then you can, this is just an example from the European Virus Archive. So this is the reason why we've got this small virus there. Usually the virus now is a blood sample. We will store it after we did a little bit of quality control, so you checked it if the hemolysis is correct, for example, or here they are doing other things to get a control. Then the sample is going to sleep in the storage. Then here down there are you, or there are researchers, there are other institutions interested in this sample. So then there's a process is organized, harmonized, how to get it. So you need this the material transfer agreement. You should ask about safety and the security, import and export permission. And we will get this information, put out the samples, and then you can send it all over the world. And just to sum up a little bit, I've got a small video. I hope it's running. We should share it. This is from the European organization.
It is always exciting to be at the beginning of a research project. However, the process of building and maintaining a collection of samples and data involves several considerations which can quickly get overwhelming. That's where professional biobanks can make your life easier. Our central facilities work according to best practices and international quality standards. Trained, qualified staff can take over tedious tasks, so you can focus on what matters most, your research. With professional biobanks, you can rely on experts. We can help you with governance issues as well as regulatory and ethical compliance. We have expertise in sample quality and pre-analytical processes, logistics, sample and data management, sample storage and retrieval. Quality matters. That's why we guarantee skilled quality management, including structured documentation. Samples are safely stored in top-notch equipment and are monitored around the clock. This provides a reliable basis for reproducible research results and successful publications based on high-quality biological samples. Professional biobanking pays off. All costs are transparent and comprehensible. Also, thanks to clear documentation and well-organized databases, managing your samples becomes even easier. Samples and data can be presented to other researchers on a European portal. When someone would like to use the samples you collected, we will ask you for permission. You remain in control of the samples and the data. Finally, professional biobanks are interconnected with researchers and other biobanks, creating many opportunities for further collaboration. All in all, we offer you maximum support so that you can concentrate on your work without distraction. So what do you say? Let's work together to advance biomedical research.
So, and I just want to say thank you. This is part of my crew, but I hope that most of you I will see tomorrow live and in person at the Helmholtz Center Munich. But I'd like to thank you for participation and really engaging this amazing community, as I say.
right, so in David's presentation, he explained why this data matters so much for a condition as rare as ours, and where our first registry ran into its limits. The good news is that we've spent the past year building its replacement, and it is live right now. Let us show you what it is and how your family can be a part of it. Choosing a registry platform wasn't a quick decision. We spent much of 2025 evaluating our options because whatever we picked would hold our community's data for years. We landed on Matrix, which we access through Combined Brain, a nonprofit whose whole purpose is speeding treatments for rare neurological conditions by bringing data together across diseases. One reason we felt confident that Matrix already runs registries for more than 200 rare disease groups across 145 countries. Going through Combined Brain also gets us the platform at a much lower cost, coverage under their research ethics oversight, and a library of standardized surveys that make our data far more useful for future clinical trials, while still being fully customizable to PURA. Here's the shift I most want you to take away. Matrix is patient-centered. Your family's data isn't just something you hand over to researchers and never see again. It works for you in 3 ways. First, yes, it contributes to research. Every profile helps researchers define the full picture of PURA and gets us closer to clinical trial readiness. But second, it helps you manage your own care. You can keep your loved one's medical information in one place and share it securely with whoever needs it, a new specialist, your pediatrician, or the school nurse. And third, it gives you insight. You can track symptoms over time, including seizures, and you can see how your own data compares with other families' anonymized data, which, for a condition this rare, is something most of you have never really been able to do. It's available in many languages, and it's connected. Your profile links to the US-based biobank, the EEG bank, and the wider Combined Brain research network. So what you put in reaches further than a single study.
We want to be straightforward about your data because trust is everything here. First, you own it. You choose to take part, and you can stop at any time. Your data is removed from the platform and from any future use. One limitation to that is that anything that's already been shared with a research before you withdraw can't be removed. But from the moment you stop, no new information goes out. And at any point, you can download everything you've entered to keep or take to a new doctor. Your de-identified information is stored on a platform built to meet both US and European privacy standards, HIPAA and GDPR. The name and the data are kept separate, so what researchers see can't be traced back to your child. Nothing is collected until you've gone through an online consent form that lays out exactly what's gathered and how it can be used. You can read it and decide. When researchers want to use the data, it goes through clear rules. PURA Syndrome Foundation acts as the steward. We manage access on your behalf, and that's overseen by Combined Brain's Institutional Review Board. An Institutional Review Board is an independent group whose entire job is to protect research participants and make sure data is used responsibly. Researchers at universities, nonprofits, and companies working on treatments can request access. And what they get is usually de-identified and grouped, so no individual is singled out. And in full transparency, when a for-profit company pays to access the data, the revenue is shared between the Foundation, Combined Brain, and Matrix. Our portion will go directly back to funding future work. There's one new term you'll run into when you enroll, and we want to make sure you understand it because it's actually a feature that protects you. It's called the CRID, the Clinical Research ID. Think of it as a unique ID for your loved one that carries no personal information, no name, no birth date, nothing identifying. All of their data gets linked under that one anonymous code. This allows researchers to safely combine and share data across different studies without ever knowing who it is. With a community as small as ours, this is how we are able to build a data set that is big enough to matter. And it means you're not starting from scratch every time. You enter your information once, and the CRID lets it travel with you instead of answering the same questions for every new study. The practical part is simple. It's free, you create it yourself at thecrid.org, and there's a short video that walks you through it with translated captions. You'll need a CRID to enroll in the registry, and the same one works for the biobank, so it's one step that opens both. Our tip is to set up your registry account first, and then create the CRID because you'll enter it as part of enrollment. Here's how you join, and there's help if you need it. You don't have to remember any of this. It's all on our website, and the QR code on the slide takes you straight there. And I believe there's also a QR code in the program. First, you create your account at pura.acrossmatrix.com. There's a not registered link right under the password box. Second, you get your free CRID at thecrid.org, and you'll enter it as you go through enrollment. And if you get stuck anywhere along the way, we've made a step-by-step family guide that walks you through the whole process. And you can always email us at support@pura-syndrome.org. We'll be honest, our last registry asked a lot of families, and not everyone made it through. We worked hard to make this one genuinely easier, and we're here to help you do it. Every profile in this registry is one more piece of the picture of PURA, and the more of us who take part, the more we give our researchers to work with. Please consider participating, and with that, we will open the floor to questions.
We want to make sure people online can hear.
So how do you make sure that the data that is put in is correct? Is it because there's no clinician involved that can corroborate what you're putting in?
David, I don't know if you have a response to this. So one thing about the new registry is that it can pull in electronic health records. So, you know, that's pulling in from clinician entered data from your doctor's visits. And when you put in things like your genetic report, something like that will be verified and made sure that it's entered correctly. But I think David brought this up in his presentation, that that's a limitation sometimes as well, so I'll let him speak on it.
So I don't know all of the details, but my understanding is that you will have the option to upload documents yourself. So although it may be that a clinician isn't entering data directly, if you have healthcare records that you're willing to upload, there is a facility for doing that. That's what I had understood. I don't know more than that at the moment, or whether this is something that they are planning to roll out in terms of clinician access. Obviously, when we built the original registry, it had a parallel arm for clinicians to essentially corroborate data that had been entered by families. But in practice, it can be difficult to engage clinicians because they're busy people, so we never wanted that to be a limitation. And I think going forward, it's really important that families have are empowered to control the data that's entered. But I do agree that corroboration of the data is paramount, because that's what essentially gives it the greatest value. But as I understand at the moment, the main way of doing that is through supporting documentation rather than clinicians necessarily linking into that account to say, yes, this is as entered and this is some additional information that that should be recorded in this record.
Are there any other questions? And if you're online, you can enter questions into the chat and we will read them aloud. No more questions? All right, well, if you think of any questions later, please don't ever hesitate to reach out to support at PURA-syndrome.org or president@PURAsyndrome.org as well. Liz and I, our doors are always open if anyone has any questions or concerns. So next,